This is Michael Altos recording part 1 of the lecture on hemostasis, taken from Berish chapter 16. We're going to start by talking about coagulation. And many of you have seen a coagulation diagram that looks something like this, full of symbols and arrows and curves and twists. And I think this is very intimidating for people. We all had to learn this at some point in medical school, but we forget it, and I think sometimes people feel guilty for forgetting it. And I'll tell you that when I went to the ASA conference a few years ago, there was a refresher course on coagulation, and the presenter said very explicitly that if you don't review this kind of material regularly, there's no way that you'll remember it. And so that's why we always start by going over the basics of the coagulation pathway. But I don't need to hit you over the head with something this complicated. People do like to make these charts as complicated as possible. They may be very accurate, but they're difficult to read and difficult to digest, and they can be, like I said before, very intimidating. My goal is to make this as simple as possible. And the reason is because we do encounter this topic all the time. Whether it's dealing with intraoperative bleeding, whether it's decision-making about lab abnormalities or invasive procedures, we do run into this subject of coagulation all the time. So my more simplified version of hemostasis looks something like this. Put as simply as possible, hemostasis is a balance between coagulation, the formation of clot, and fibrinolysis, the breakdown of clot. And if we can look at everything in coagulation in this context, I think we're off to a very good start. Unfortunately, I don't think the Berish chapter does a very good job of covering these issues in a clear and appropriate manner. So I've tried to do my best to distill the material, to add to it when necessary, and give an organized presentation. Let's start on the coagulation side. Coagulation can be broken down into primary and secondary hemostasis, where primary hemostasis primarily involves the platelets, and secondary hemostasis involves other clotting factors. So let's talk about primary hemostasis, which is basically platelets adhering and becoming activated at a place where the endothelium has been disrupted. And there are 4 components that we need to discuss. There's the adherence itself, then the activation of the platelets. Number 3 is stabilization of that platelet plug. And we should also discuss inhibition, the checks and balances that undo some of this platelet adherence and activation. So first we'll talk about platelet adherence. When there is an injury to a vascular wall, platelets attach to that site of injury. We can see that here is a vascular wall, here's the endothelium, here is some denuded endothelium, there's the vascular injury, and these various receptors, these GP receptors, glycoprotein receptors, are here, and platelets adhere to these various receptors using various attachments that exist on the platelet already. And so this is adhesion. The platelets are particularly interested in attaching to substances like collagen, fibrinogen, and fibronectin. And so these are things that the platelets are looking for when adhesion starts. They also attach to von Willebrand's factor. Here's another picture showing the same thing. Here's a platelet sort of hanging out and rolling along an endothelial surface. There's collagen, there's von Willebrand's factor, and the platelet encounters an area where there's been damage. And immediately the glycoproteins on the platelet surface bind to the collagen. The platelet, as we'll see in a moment, starts to change its form and become more stabilized, and this process will be discussed more on the next slide. So when platelets become activated, what that means is that there are G protein coupled receptors within the platelet, and they cause the platelet to release all sorts of different substances: thrombin, ADP, thromboxane, serotonin, epinephrine, vasopressin. Phospholipase C is also activated as the platelets adhere. G protein coupled receptors also cause this to be activated. And as the ADP and the serotonin and calcium are released, the platelet starts to activate more platelets, and the platelets start to change shape. And this is just a cute little diagram of a platelet with all sorts of granules inside of it containing these substances that we just discussed. And so now back to the big picture, we get to the third step, which is platelet stabilization or aggregation. And we see our platelet has now changed its form as a result of stabilization. And as the glycoproteins become activated, we start to get this very complicated binding of fibrin and von Willebrand's factor. We get cross-linking between other platelets, and this really stabilizes the platelet plug. We should also keep in mind that, like every system in the body, there are checks and balances, and there are inhibitory processes going on at the same time. So the platelets contain substances like nitric oxide and prostaglandins, or rather, those substances are secreted by endothelial cells, and they inhibit thromboxane, and so they work against some of these processes of adhesion and aggregation and stabilization. These cause vasodilation and platelet inhibition, and so the process is always in a state of flux, where the platelets are encountering a site of vascular injury and beginning this process, and meanwhile nitric oxide and other substances are working to keep this process in check. So take a moment if you have any questions or concerns that you need to discuss in class. Jot them down and we'll be happy to go over them, or you can email me or track me down and we can discuss this in more detail. So we've discussed primary hemostasis, which involves the platelets. Let's move on to a quick overview of secondary hemostasis, which really just means using clotting factors to create a thrombin plug. So if platelets are the main player in primary hemostasis, the thrombin plug is really the main player in the secondary hemostasis. And once again, we have the steps of activation, amplification, and propagation, as well as an inhibitory component that keeps the system at balance. I have reproduced one of the diagrams from our textbook, and while this does look like one of those complex diagrams full of arrows and dotted lines, I think this one actually does a pretty good job of conveying useful information, and so we're going to go through it step by step. The first part of the process is activation or initiation, and this involves the arm of the coagulation cascade that we learned about in medical school as the extrinsic pathway, the one that focuses primarily on factors 2, 5, 7, and 10. The first player in this pathway is always tissue factor. Tissue factor is a protein which is membrane bound, and it's found in vascular endothelial cells. And this protein is just waiting for an injury to occur, at which point it becomes exposed to blood, and this sets off the cascade. So we see here tissue factor, and when it's exposed to blood, it encounters factor 7 and activates it to factor 7A. The complex of tissue factor and factor 7A together then become their own enzyme, which is able to take factor 10 and convert it into factor 10A. You can see here it also can convert factor 9 into factor 9A, but we will leave that out for the moment. But the tissue factor 7A, 10A complex then becomes its own enzyme, which is able to encounter factor 5, as well as doing some other things. And ultimately, the tissue factor 7A, 10A, 5A complex can find factor 2, also known as thrombin, or actually prothrombin, and activate it to become factor 2A, which is thrombin. So if we had to summarize the activation pathway or the extrinsic pathway, it would be tissue factor involving factors 7, 10, and 5 to create activated thrombin. The second step in secondary hemostasis is amplification, and this involves what we learned as the intrinsic pathway. And basically it takes advantage of the fact that we have just created activated thrombin. And thrombin does many different things. 4 things we want to highlight in particular, shown over here with these red arrows. Thrombin is able to further activate platelets. Thrombin can activate more of the factors 5, 8, and 9. Thrombin is able to release factor 8 from its factor 8 von Willebrand complex, and we'll talk about that again a little bit later. And it also activates factor 11, which in turn comes and activates factor 9. So thrombin really causes a burst. It's called a thrombin burst. Thrombin really makes this pathway explode with activity. And once we have factor 9A and factor 8A, that can activate a lot of factor 10, which leads to more factor 2A, and the system is really in high gear. This is the amplification step of secondary hemostasis. Just looking at the diagram over here, we see thrombin specifically activating factor 10 into 10A, activating factor 8 by releasing it from von Willebrand's factor into factor 8A. We see here's factor 11 going to 11A, which activates 9 into 9A. And then here's the 9A, 8A complex together, called the tenase complex. Why is it called tenase? Because it cleaves factor 10 into activated factor 10A. And 10A plus 5A that we had lying around from before becomes the prothrombinase complex, which makes a lot more activated thrombin. And now thrombin can go to work making a very useful and stable clot. Propagation is the third step, and this involves fibrin. The thrombin is very nice, but the thrombin is really just the infrastructure. It's really just the scaffolding to allow fibrin to come and make a dense and stable fibrin plug. So the platelets provide the surface for the clot to be activated. Thrombin, factor 2A, activates fibrinogen, which is the precursor to fibrin. It also activates factor 13. And together these allow you to make a stable fibrin plug, which is really the ultimate goal of secondary hemostasis. We're going to spend just a moment and discuss the inhibition component of this pathway. There are many different players here. One of them is called tissue factor pathway inhibitor. And this leads to an interesting question, actually. We saw before that the tissue factor 7A complex can activate factor 10A. So then why do we need factor 8? We're just going to look back for a moment. And here we see factor 8 mixed with factor 9 making all this factor 10, and then things really get exciting. But what do we need that for? We saw over here that this substance right here can also activate factor 10A. In other words, why do hemophiliacs bleed? Hemophiliacs have an absence of factor 8 or factor 9. And the question is, well, who says they need it at all? It looks like everything that this thing does over here is accomplished over here as well. And the answer is because tissue factor pathway inhibitor regulates this step right here. It's a negative feedback loop. So as soon as we have some factor 10A, factor 10A binds to tissue factor protein inhibitor, and that inhibits this process. So this process is really very muted. And for further coagulation, for the process to really get underway, you need factors 8 and factor 9. And that's why hemophiliacs bleed. It's not that they can't make any activated factor 10A, but the process shuts itself off. Interestingly, heparin stimulates the release of tissue factor protein inhibitor and potentiates it. Other sources of the inhibition component would be thrombin and antithrombin 3, also called AT3. Antithrombin 3 inactivates a whole bunch of different components. Not just thrombin, but also many of the factors, including factor 7A, 9A, 10A, 11A. And this is important to remember because heparin acts at antithrombin 3 by binding to it and potentiating it. That means it makes it more active. And this is an important concept that we'll come back to a little bit later. A few other inhibitory mechanisms. So there's protein C and protein S. These are vitamin K dependent factors that exist in the system. So we know that thrombin binds to a protein called thrombomodulin, which is in the endothelium. And when thrombin binds, then is able to bind to protein C and create a complex called activated protein C. And activated protein C binds to protein S, and that whole substance together can start chewing up factors like factor 5A and factor 8A. So we see we have a system in place which is started out by thrombin, which creates clot, and this system ultimately starts breaking down clotting factors. Finally, there's a substance in our body called heparin sulfate, spelled differently than the heparin drug that we give. And this heparin sulfate actually occurs naturally at the blood endothelial interface. Your body creates this substance, which is a natural anticoagulant. So your body is able to create a number of different substances and mechanisms to break down any clot that's being formed in the body. I'd like to finish this section by going over a few additional points about clotting factors. I just want to remind you that all of the clotting factors are proenzymes, as we've been alluding to all along. So that means that they aren't active until something happens to them. They're also, and a serine protease is basically the mechanism here. So these enzymes are cleaved chemically and they become active. They're all generated from the liver except factor 8, which is generated from endothelial cells. Remember that factor 8 starts out bound to von Willebrand's factor, and thrombin cleaves the two from each other. And so that means that a person who just has a factor 8 deficiency has hemophilia A. But a person who has von Willebrand's deficiency actually has a platelet problem, but he also clinically has hemophilia A because he doesn't really generate any factor 8. I want to remind you that von Willebrand's factor is synthesized in endothelial cells as well as megakaryocytes. You should remember that the vitamin K dependent factors are 2, 7, 9, and 10. And we need vitamin K in order to synthesize those factors and in order for them to function. These are important concepts that could be tested on a board exam, and that's why I like to highlight some of this trivia on this slide. You could also be asked which factors have the shortest half-lives during storage, and the answers would be 5, 7, and 8. These factors are the first to break down during storage of plasma. We've covered quite a bit there, and so we're going to pause for a minute for you to review the material, jot down any questions that you might have, which we can discuss in class, or you can contact me ahead of time. And then we're going to move on to one last little section here before we end this recording. So far, in our very simplified diagram, all we've talked about is coagulation. But there is another half of this equation, which is the fibrinolysis. So we've talked about how to create clots with platelets and with coagulation factors. But our bodies also have a way to break down clots, and that would be fibrinolysis. Fibrinolysis, very simply, is the process of dissolving clots. And the key player here is plasmin. Plasmin starts also as a proenzyme called plasminogen, and it circulates and it binds to the fibrin clot. And the plasminogen is converted, it's activated into plasmin. How does this happen? Well, your body has a substance called TPA, tissue plasminogen activator, and that converts the bound plasminogen into plasmin. So here we have a picture of a nice fibrin plug, a nice clot, and here's plasminogen sitting here, and here comes some TPA released from the endothelial cells, and it activates plasminogen into plasmin, and plasmin starts chewing up this clot. We also have exogenous plasminogen that we can give, or rather exogenous activators of plasminogen, so streptokinase and urokinase and even recombinant TPA. And when we inject that, it activates actually not just the bound plasmin, but any circulating plasmin as well. And that's why we get such a widespread breakdown of clot when we give TPA as a drug. Now your body also has a substance called PAI, plasminogen activator inhibitor, which binds to the TPA and accelerates its clearance so that it can't bind to plasminogen, activating it into plasmin and breaking down clot. Instead, that process is inhibited, and that's why it's called plasminogen activator inhibitor. So the checks and balances continue on this side of the equation as well. There is a substance called thrombin-activated plasmin inhibitor, and that's another substance that binds to TPA and inhibits it from activating plasmin. Now let's talk about plasmin itself. Once the plasmin finally gets activated, its job is to break down fibrin threads, to break down these fibrin clots. And when it does that, it creates byproducts called fibrin degradation products or fibrin split products. What happens to the plasmin after it has done its job? Well, plasmin can be attacked by a substance called an antiplasmin, and that will degrade the plasmin. And so excess plasmin circulates through the body. It starts degrading coagulation factors, especially during disseminated intravascular coagulopathy, DIC. And so the antiplasmins are yet another check and balance for plasmin that circulates through the circulation. At this point, we've covered quite an awful lot of basic science, so we're going to stop here. Please let me know if you have any questions or come prepared to ask them in class. And in the next section, we'll move on from the basic science and start talking about more clinical aspects of coagulation and hemostasis.